Hello, my name is Haley Beckler. I'm a student here at Mayville State University. My research is polymeric scaffold with wheat brand Arabinoxylan for tissue engineering. This is my poster, and I will go into each topic in more detail on the next slides. Arabinoxylan is a heteropolysaccharide that consists of a linear xylose backbone with variable arabinose substitution. It is also cross-linked with ferulic acid. It is the primary hemocellulose found in the cell walls of plants, along with wood and cereal grains like wheat bran, which is what we extracted it from. The purpose of this research is to create a three-dimensional scaffold for tissue engineering by utilizing the arabinoxylan. In current medical research, live subjects are being used to perform various tests, which puts their lives at risk. This can cause a huge ethical dilemma, and that is why scaffolds could be greatly utilized. When preparing a scaffold for tissue engineering, there are several considerations that are important for designing it for suitability, such as porosity, mechanical strength, biodegradability, non-toxicity, and cell adhesion. 3D scaffolds need to have specific mechanisms because they do not have the ability to access a large surface area because they are 3D. In the image you can see on the slide, it is of chitosan in a scaffold. Chitosan is a viable biomaterial for tissue engineering because of its multifunctional structure and because it modifies the properties of the scaffold, and that is why we are also utilizing it in the research. The basis of this research is arabinoxylan. In this research, we extracted arabinoxylan from wheat bran by combining it with 4.5 potassium hydroxide. This mixture was heated for 2 hours, centrifuged for 2 hours, and then the superheat was kept. That was combined with ethanol, kept in the fridge, and then centrifuged again. That was then washed with distilled water, placed in the vacuum oven to dry, and then placed in the freeze dryer to lyophilize. The pellet that was formed was then dissolved in distilled water, and this image is our arabinoxylan solution. For the experiment, various concentrations of arabinoxylan were mixed with chitosan and polygactronic acid separately. The volume of solution in the tubes remained 20 milliliters for each solution. The concentrations started at 10 milliliters and 10 milliliters, and then when one would decrease by 2 milliliters while the other would increase by 2 milliliters, and vice versa. There is another concentration for arabinoxylan and chitosan, which I will talk a little bit more about in the next slide. The solutions were added together by slowly dropping one of the solutions by pipette into the other solution to allow them to mix thoroughly instead of one sitting on top of the other one. And then once the solutions were mixed, they were sonicated to mix even more. So like I talked about in the last slide, there is an extra concentration for arabinoxylan and chitosan, and that is because if you look at this first image on the slide, you can see how that solution has such good viscosity, and the bottom image shows the comparison between the mixture with chitosan and the mixture with polygactronic acid, where the acid is so much less viscous than the chitosan. Viscosity is a key quality in the creation of scaffolds because it allows the cells to actually have the ability to adhere and grow up the scaffold to create a more three-dimensional structure instead of a two-dimensional structure. Because this research just started to take off, there's a ton of opportunity for continuing research plans. Scaffold will be constructed with the different concentrations of the made-up solutions talked about previously, by centrifugation to discard the supernatant and then freeze-dried to absorb more moisture. The scaffolds will then be tested for porosity, mechanical strength, biodegradability, non-toxicity, and cell adhesion to conclude which concentrations will best suit animal cell life, and then those scaffolds will be used in cell culturing.